Good morning, good afternoon, or good evening, depending on where you're at in the world. On behalf of the International Association of Bloodstain Pattern Analysts, I want to welcome you to the Scientific Foundations of BPA webinar. I'm your president, Celestina Rossi, and I would like to introduce today's speaker. Jeremy Morris is a certified bloodstain pattern analyst through the International Association for Identification. He's a technical leader in BPA and trace evidence examination at the Johnson County Sheriff's Office, which is located in Kansas, where he's worked for over 20 years. He's a member of several forensic organizations, including the Midwestern Association of Forensic Scientists, the International Association for Identification, the American Society for Trace Evidence Examiners, and of course, the IABPA. Jeremy's the editor of the Journal of Bloodstain Pattern Analysis, as well as the Journal of American Society of Trace Evidence Examiners. He's a past member of the BPA subcommittee of the Organization of Scientific Area Committees, better known as OSAC. We had an outstanding presentation this morning with approximately 350 attendees. I think this afternoon's webinar is going to beat that. And the attendees were from around the world. You guys will have the opportunity to ask questions in the question answer section. However, what we learned from this morning was participants were asking questions about such things like the NAS report, error rates, methodology, and Jeremy covers those extensively in the presentation. So if you don't get your question answered after the presentation, we will provide email addresses and contact information that you can ask the executive board or the conference committee. However, the executive board and the committee will be in the audience, and they're going to help answer any questions that you may have. Again, I ask that you direct those to the question and answer session and not in the chat area. If you need a certificate for this webinar, you'll have to answer yes to the poll at the end of the presentation. A recording of this presentation will be provided for 7 days. It's available for 7 days, and we are going to try to post that on our website. If not, it's going to be available by email. A Spanish translation for this presentation is available. There's a button at the bottom of your screen. For that, there's also a button on the home screen right now. And last but not least, the registration for the IABPA 2020 virtual conference, which is going to be held next month, is now live on our website. And so if after today's presentation, you are interested in being part of a 40-hour conference, then that cost is $30 for the entire week. We would love to have you. And that is available on our website at www.iabpa.org. Without further ado, I would like to give you Jeremy Morris.
Thank you, Chelly. And I pass on my greetings and welcomes to everyone around the globe. Good morning, good afternoon, good evening, wherever you are at. And first and foremost, I want to thank the IABPA for providing this opportunity. I also wish everyone good health during these times. These are things that we appreciate more and more. Again, my name is Jeremy Morris. I'm with the Johnson County Sheriff's Office Crime Lab. And I'm glad that we're all having this opportunity today to talk about the scientific foundations of bloodstain pattern analysis. So we have a key question that has been asked a lot recently, and this is the question: is bloodstain pattern analysis a valid and reliable science, or is it just a different form of junk science? This is a critical question in all aspects of forensic science, especially when we start transferring the information into courts of law. So we're going to address this question in several different ways. We're going to give a brief description of bloodstain pattern analysis, discuss methodology, what is a scientific method, what makes a method reliable. Can reliable bloodstain work be useful in criminal investigations? We're going to do some discussions about why errors occur in science and how we can really figure out what the source of that error was. And then also discuss what makes an individual competent and qualified in BPA. Along the way, as Chelly mentioned, we're going to have a rather extensive discussion about how we can determine, or if we can determine, the potential error rate. So before we get into that, it's good for us to have a discussion about two different kinds of conclusions that are offered in forensic science. And those are source conclusions as well as event conclusions. And source conclusions are trying to associate objects with people. And forensic science does that very well. So let's consider this scenario where we have a homicide and a gun was used in the homicide. We collect that gun, and from the gun, we swab the front face of the trigger in the hopes that we might get a DNA profile. We take that swab, we do some examinations on that swab and develop a genetic profile from the swab. That profile is then compared to persons of interest, and we make an association of that swab to a specific person. And here, this would be the accused or the suspect. So here we've made a source conclusion. But the obvious follow-up question is this: how did that DNA get there? Yes, we've made an association, but oftentimes it's not whose DNA that is the critical question. It's how did that DNA get there? I asked my biology colleagues if they ever get that question, and one of them said, we get it all the time. You know, the source of the DNA is not under dispute. What is under dispute is the event. How did that person's DNA get there? So if we can consider this scenario a little further, if the most natural explanation for how this person's DNA got on the trigger of the handgun is that that person pulled the trigger. Well, if I am the attorney for this client, I'm going to say, no, that's not how the DNA got onto the trigger. There are other ways, and this is actually how my client's DNA got onto that trigger. My client remembers shaking hands with someone who was wearing a pair of gloves. And we know that handshaking transfers DNA onto a surface. So my client's DNA was transferred onto that glove. And then that person is the one still wearing the glove who pulled the trigger, because my client is really a saint and who would never commit this kind of crime. So this here is a source conclusion. When it comes to blood, we can extend this even further, or look at it more specifically. Here we have two red-brown stains that are on a sock. They visually look like blood. So we'll take cuttings of the sock, we'll do some extractions and some analyses, and again, we're able to make a source conclusion that the DNA from the cuttings of the red-brown material on the sock are associated with this person. But again, the most natural question is to ask, how did the blood get on the sock? Because there could be all sorts of reasons. There could be very innocuous and non-violent and very innocent reasons why a person's blood got onto a sock, or there could be some criminal reasons dealing with criminal activity. DNA cannot answer an event conclusion question. They just can't do that. They're outstanding at providing source conclusions and answering those questions. But answering the question of how did the blood get there is outside of the realm of DNA. And to my knowledge, the only discipline that's able to answer the question that deals with event conclusions for blood is bloodstain pattern analysis. And there are times where that question, how did the blood get there, is absolutely essential to the case. So to throw out bloodstain pattern analysis totally means you have no ability then to answer that very critical event conclusion question. So what is bloodstain pattern analysis? There's a couple of different ways we can explain that or answer that question. We can give a long technical definition that it's the examination of blood at a scene or objects from the scene to identify the activities or mechanisms that produce those patterns. But one of the ways that I like to explain it is that the blood stains tell a story. Those blood stains tell a story of things that happened after someone was injured. Now, it's a partial and fragmented story, It's like photographs of an event rather than a movie of an event, but it is a story. So when I go to a crime scene and I examine the bloodstains, my job is really to read the story of the bloodstains. What story are those stains telling me? Ignoring the stories that everyone else is telling me, but what do the bloodstains tell me? So here's an image, and there's a number of different bloodstain patterns here, and we can see that those bloodstains tell a story. They tell a story of this location in a moment of time and something that happened. So we need to read that story. And we do that by looking at the features of the bloodstains. This slide has 12 different pictures on it, 12 different bloodstain patterns. Whether you have training in bloodstain pattern analysis or not, you ought to recognize that these patterns all look different. The reasons why they look different is because they were created using different mechanisms. And those different mechanisms are different parts. And that would be the classification side of things. Classification is this pattern was created by this mechanism. That could be an impact mechanism, a transfer mechanism, it could be an expiration mechanism, but those are classifications of those bloodstains. But other times we want to know, what does that mean? Yes, there's an impact pattern, but what does that impact pattern mean in the context of this scene? So analysts routinely can determine other aspects. These are reconstructive aspects of the scene. Where were people at a scene? How did they move throughout the scene? Is there a point of origin in two-dimensional or three-dimensional space that we can say that this pattern was created at this location? Can we get an idea of the potential weapon that was used? Or if we have identified a weapon, can we determine the minimum number of impacts, blows, shots that took place? There are times where we may be asked, is the suspect injured? Is there a potential that the suspect is injured? Or is there the possibility that bloodstain patterns can be on the suspect? I have considered all of these scenarios at some point in my career. And even though BPA can provide these types of conclusions, my ability to provide any of these conclusions is entirely dependent upon the quality and the quantity of the bloodstains present at the scene. There have been scenes where I walk in and I simply cannot provide a lot of information because there's not a lot of bloodstains present, or the quality of those bloodstains is not sufficient to tell a significant portion of the story. So going back to this image again, the questions may be, where was the victim? Was the victim standing up as the suspect is saying, or was the victim on or near the ground? How many times was this person struck? Was the bat involved, or was some other weapon involved? What were the actions of the individuals after these events? Bloodstain pattern analysis has the potential to answer some of those questions. Now that's good and noble, and we have a lot of aspirations for BPA, but just because we have these noble aspirations doesn't mean that that approach is scientific. If this approach is not scientific, it is of little value to the investigators or little value to the court because the process is likely not trustworthy. So let's look at BPA and determine, are there scientific foundations that bloodstain pattern analysis rests on? And this is becoming more important today. In 2009, in the United States, the National Academy of Sciences issued a report on a review of the state of forensic science in the United States. And it covered virtually every discipline of forensic sciences, and essentially said, these are things that are going well, these are things that need to be improved. In the section for bloodstain pattern analysis, the last sentence of that section reads as, quote, "The uncertainties associated with bloodstain pattern analysis are enormous." when that sentence is taken out of context, it looks really, really bad. But when you read it in context, it's not as damning. Now, it's saying at this time, there are things that bloodstain pattern analysis needs to work on. But since 2009, there have been a number of different articles that have been written about BPA, or podcasts that have been given, or other avenues that have accused bloodstain pattern analysis of not being valid. It's been called junk science. It's been called dubious. It's been called bogus. So it's been challenged. And I'm actually going to say that these challenges are a good thing. Now, that may seem crazy. These are pretty aggressive challenges to the work that I do, that I put time and effort in. How can this be a good thing? It is a good thing because it provides us the opportunity to examine the discipline and to question whether or not it truly is scientific, and to provide answers to those challenges. And we are studying this in all sorts of ways. We study this in our introductory workshops. We study this in our research facilities at universities, and some of you are studying it right now. I intend to do this earlier, but I want to give a shout out to Professor Valentin's class at the University of New Haven. We have a class this is what their course is on right now, is bloodstain pattern analysis. So this is critical to what they've paid for. Is this a valid science? So for the students of Dr. Valentin's class, welcome, and I hope that this helps you in your studies. So here we go. What are the scientific foundations for bloodstain pattern analysis? The first thing that we need to do is answer a common criticism. Herbert McDonnell was a large figure and is a large figure in the history of bloodstain pattern analysis. But bloodstain pattern analysis was not created by Herbert McDonnell. Herbert McDonnell in the 1970s and 1980s played a large part in bringing bloodstain pattern analysis to prominence, to demonstrating its usefulness in criminal and civil investigations. But the work that was done on evaluating bloodstain patterns went before him for 100 years. Going all the way back to 1863, we start seeing published research about scientists and doctors and investigators who were looking at these bloodstains to try and determine what stories could be told. This slide contains a very short list of some of the papers and some of the work that has been done historically. And you can see all of this work was done long before Mr. McDonnell came onto the scene. If you want some more information on this, you can read Bevel and Gardner's book, chapter 1, that goes through the history of BPA, taking a historical look at it. But what are the foundations? To my knowledge, right now, you cannot go to college and get a degree in bloodstain pattern analysis. You can get a degree in forensic science, but you can't get a degree in bloodstain pattern analysis. But that doesn't mean it's invalid. What we find is we study bloodstain pattern analysis is that it draws from a broad range of classical sciences. These classical sciences that are well established, that are recognized, that are respected, have principles and components that directly influence our ability to analyze bloodstains. So if we take a quick walk through them, regarding anatomy and physiology, it's important for me as an analyst to understand, to some degree, the circulatory system, to know the difference between arteries and veins, to know which arteries and veins are major and which ones are close to the surface and more likely to create certain bloodstain patterns, which arteries and veins are under the surface. The respiratory system is also important because an expiration pattern is a bloodstain pattern that is created by blood being expelled from the nose, mouth, or wound to the respiratory system. We need to understand the composition of blood. And it's also important for us to understand wound location and dynamics, to know the difference between an incise wound and a laceration, to a contusion, to a bruise or a scrape. We need to understand all of those and apply those concepts to BPA. When it comes to chemistry and biochemistry, when we go to crime scenes and we search for bloodstains, we use various reagents. We need to understand the chemistry of those reagents. We need to know what they're looking for. Are they looking for proteins? Are they looking for fats or salts? When we use them to search or enhance. When it comes to our presumptive testing or other serological reagents, to understand that chemistry, to know the enzymes that are being looked for in blood or saliva, and also to understand the blood clotting process. Understand why blood clots, how long it takes, and are there any things that may cause that blood clotting process to slow down or to speed up. We're going to take a little bit of time to talk about physics and fluid dynamics, because this is absolutely essential for us to understand these concepts, because they play a very large role in what we see in the static patterns. The general concepts like momentum, acceleration, and kinetic energy are important. Newton's laws of motion have direct application to blood and flight. And then these fluid concepts, static fluids or fluid in motion, dealing with surface tension, viscosity, drag, adhesion, cohesion. All of these have been well studied outside of BPA. And here are just a few examples of some of the papers where the researchers were studying properties of fluids, not specifically looking at how those fluids deal with BPA, but we can apply those concepts directly. On the left-hand side, the study deals with the spreading of a fluid as it strikes a surface and it generates droplets. This would be the splashing mechanism that they discuss. We see that in BPA. The center discusses the concepts of how a fluid, when it strikes an inclined surface, a droplet of a fluid striking an inclined surface, how it spreads, not just lengthwise, but also the width, and what factors affect that spread, and can we predict that spread mathematically? And then on the far right, we have a discussion or a study about jets of fluid and how those jets break up into smaller droplets. That has direct application to projected patterns. Additionally, the screen printing industry or the inkjet industry wants to know how blood interacts when it strikes, or not blood, how fluids interact when they strike textiles. So there are other studies that deal with that. As a droplet of a liquid strikes textile and spreads across, how does it interact with that textile? Does it penetrate the textile? And what does that look like? So we have this study that deals specifically with that. So here we have fluid dynamics that deals specifically with fluids other than blood, but yet we can pull those concepts and make a direct application to bloodstain pattern analysis. We also have concepts of mathematics that we use in BPA. These concepts that we use for area of origin, the angle of impact, areas of convergence. We use math for those. We use trigonometry, and we use those very well. We have studied these concepts extensively. When it comes to logic, there's inductive and deductive and abductive reasoning. Are there valid approaches to reasoning and logic? What about Bayesian approaches towards event reconstruction? There are rules to this, and we can apply those rules to how we interpret bloodstains. So we also need to follow a specific process, and that's a critical question. What process are you using? Is your process like throwing a dart, where you're just blindly throwing a dart and it is what it is, and you're not really following a method? Or do you have a defined method? If it is a defined method, then that would be you have a starting point and you have various steps of which you follow. Those processes, as you can see, are completely different. The scientific method is like the second. It is a very defined process that can be explained in a number of different ways. Some people consider it a linear process that has starting with a question and ending with an answers. Or it can be a circular process where you start, you get some information, and then you determine that you need to get more information regarding that. So you start all over. So BPA needs to follow the scientific method. There have been at least 5 different papers that have been published regarding the scientific method in bloodstain pattern analysis. Fortunately, all of them have been published in the same journal. So if you go to the Journal of Forensic Identification, you can see those papers. And each author here takes a different approach, but all of them are trying to demonstrate that BPA does follow the scientific method. Now, for me, when I'm asked, what is your method, Jeremy? What method do you use in bloodstain pattern analysis? Here's my method. It has 4 steps: documentation, classification, analysis, and peer review of conclusions. Those are the steps that I use. And for me, it's important that I have an established method that I follow all the time. And not only that I follow, but that I can explain to others that this is what I do to get my answers. Starting with the features, coming up with partial answers, getting some additional information, coming up with conclusions, and then almost the most important part is having another analyst review those conclusions. So this is my methodology. For those skeptics among us, I welcome you, which may seem rather strange, but I do welcome you among us. And one of the things that I would ask for you to do is when you look at the mistakes that have been made in the past, and people have made mistakes in BPA, to determine what method were they using. Were they following a method, or were they just kind of coming up with stuff on their own? And we're going to emphasize over and over again that reliability, in order to be reliable, it is essential that we have a defined method. So here's the pathway of the methodology for bloodstain pattern analysis. We start with observations of the bloodstains, the size, shape, distribution, appearance, and location of stains. We take those features and we try and from those features come up with conclusions. Those conclusions can be what mechanism created it, and also what does that mean in the scope of the scene. While that may be my approach and my method, is this approach scientific? Is it really based on scientific principles? I want you to look at this picture. What do you see? For most of you, the immediate answer is I see a zebra. I'm going to make the point that you have given me a conclusion, that is a conclusion based on other observations. So why don't we look at it this way? What do you see? For me, I see a 4-legged animal that has a long neck, an elongated head, that it has a mane, the face is black, the body and neck and legs have alternating stripes that are black and white colored, and those stripes are random. And it visually looks like another animal that I recognize as a horse. Those are the observations that we use almost without thinking to come to the conclusion that it's a zebra. But the conclusion is based on observations. When we apply this to BPA, what do you see? For trained analysts, you can look at this picture and almost immediately say, I see a drip pattern. But again, that is a conclusion. The features are what you really see, and then you interpret that into a drip pattern. The features are a large irregular shaped central stain. That central stain is surrounded by several smaller stains. Those smaller stains are elliptical to circular in shape. The larger of those smaller stains are towards the middle. As you move further away, those smaller stains get smaller and smaller. They all seem to radiate out from the central stain. In the lower right-hand corner of this image, I see an absence of those smaller stains. Those are the observations that justify the conclusion or interpretation that this is a drip pattern with a void in the lower right corner. In both situations, the observed features were used for the classification. But is that approach scientific? We're going to call this feature-based classifications. Is this used anywhere else? And it absolutely is. We're going to study or briefly discuss other sciences that use features, visual features, to classify things. And we'll start with biology. We started with the zebra, we'll continue with that. Biological classification that began with Linnaeus was originally based solely on visual features, trying to take this vast number of plants and animals and organize them in a certain way to show various relationships. And that taxonomy, that classification to get from kingdom and phylum and order and genus and species is based or historically based on visual features. Now, currently it uses additional things like genetics, but it still largely rests, at least initially, on visual features. So here we see this cell. We start breaking it down. Is it one cell? Is it several cells? If it's one cell, if we look internally, are there organelles, or is it a mass of things? Is there a cell wall along with the cell membrane? And all of these visual features that help us move through this classification to eventually get to the final step of this is the genus and species that we're looking at. Another group of people that uses visual features for classification is pathologists, specifically those who are determining the pathology of tumors. The initial screening and classification of these tumors is based on the morphology or the shape of those cells. Let's say my doctor finds a mass in my intestine, and they want to know, is that mass cancerous? They'll take a sample of that mass, send that sample to a pathologist who will create a microscopical slide. And then they will view the cells and look at the visual features of those cells to determine if those cells are healthy cells or if those cells are cancerous cells. And if it is cancerous, what kind of cancer are we looking at? If it was lung tissue, is it small cell sarcoma? Is it large cell sarcoma? Is it something different? And although they currently use other techniques in addition to visual features, the visual features are the first step towards that classification. Archaeology also uses this. This is something that I wasn't aware of until recently, that if we have an archaeological dig and we find pottery, archaeologists can use the features of that pottery to classify that pottery into specific geological or archaeological time eras. Is it early Bronze Age? Is it late Bronze Age? Is this pottery now moving into the Iron Age? And they will use that pottery and examine the visual features of the pottery to classify that. Now, this is going to be a relative dating technique. But if there are times where they can't do absolute dating techniques, the relative dating technique is used to determine what age that specific layer of debris came from. And it's using the visual features of the pottery. The same thing is done with arrowheads, trying to look at these arrowheads and the design of the arrowheads to try and link them into a specific time frame or age. So what we see is that feature-based classifications is absolutely used numerous times outside of bloodstain pattern analysis. It's used routinely in sciences that we recognize, in sciences that we trust. So using features to classify something is a valid approach. But what is important for us is to understand what is driving those features. In biological classification, as well as in the pathology of tumors, the driving force is genetics. You change the genetics, you change the species. You alter the gene, you're going to alter the proteins and cause that normal cell to become a cancerous cell. In the dating of archaeological artifacts, the driving force is cultural trends. In bloodstain pattern analysis, the driving force is the forces that are involved, forces that act upon liquid blood, as well as the surface features. So if we go back to this slide that we've seen before, looking at these 12 images, all 12 of these look different because different forces are involved. If I have a given amount of a liquid blood and I expose it to different forces, it's going to react in different ways and cause different appearing patterns. The more we understand the forces involved, the better we understand why bloodstains look the way they do. This is why understanding physics and fluid dynamics is so essential to bloodstain pattern analysis. But now let's move to the second category of conclusions. That second category is event reconstructions. And it's, okay, this is an impact pattern, but what does it mean? Yes, I see that blood was struck, but what does that mean? These can be the reconstructive conclusions that are made, that we discussed about people moving within a scene, the sequence of events, or the position of people. Were they standing up, or were they kneeling, or were they on the ground? What are the scientific foundations for these kinds of conclusions? And perhaps more importantly, are those kinds of conclusions used anywhere else outside of bloodstain pattern analysis? Does anyone else try and use things to reconstruct stories? And the answer to that is yes. Now, this is an example that has been used in crime scene reconstruction as well as bloodstain pattern analysis. It's Nicholas Stenou's principles of geology. What Nicholas Stenou discovered is that as we look at geographical features, certain things become obvious, that the layers of rock on the bottom are older than the layers of rock on the top. So we have this principle of superposition. And then if you have a layer of rock that is continuous for miles and miles, you know that that rock was deposited at the same time. Even if that layer of rock was broken up, there's a gap in it. And so there's various principles of geology from Nicholas Stenou that deal with reconstruction of things. But there's also more than that. Let's go back to archaeology. So we go back to archaeology of ancient cities. What we've found is that we can go to an archaeological site and we discover that there are multiple cities that are built on top of each other over time at this location. That numerous times a city has been built, it was destroyed, and then another city built on top of it. So we get these layers, or these various strata. And as archaeologists dig through that strata, we can apply the sequencing to it. The lower layers are older than the upper layers. But there's also other things that they can look at. If we look at a specific layer, we find a heavy layer of ash. That can demonstrate how that city was destroyed, that perhaps that city was burned by fire. Looking at the kind of pottery that's there to get an idea of the ethnicity of the population. We can also use the remains of the animals that were there, that were eaten. If we are looking at the trash areas and where the bones of animals that were eaten were thrown away, if we don't find any pig bones, that gives support that this was used by Hebrews. We can also determine the economic status of individual households and of the city. Look at a house. Is it a smaller house? How many rooms does it have? We can look at the number of homes within that specific layer and how many of those homes have multiple rooms, or was there one prominent individual within that city? We can look at the things that are found within a room and determine the purpose of that room. Was it a storeroom? Was it a bedroom? Was it a living area? Is this where the household animals were kept when they weren't outside? We can look at the influences of other cultures and other cities. If we can see their influence on the pottery or on the architecture, or we can also look connecting a specific layer with a specific time frame of another nation. Let's say we are at we've dug down through 4 layers, and we get to the 5th layer of this city, and we find a coin, and on that coin it identifies the king of a nearby nation. Well, what we can now do is we can now link that layer to the known time frame of that other king. So this is element-based reconstructions with archaeology. And one that is really fascinating to me is this field of failure analysis. And this is really, really important in civil litigation, where an item broke. And why did that item break? It failed. Did it fail because it was a design flaw? Did it fail because it was a manufacturing flaw? Or did it fail because the user misused the product? And failure analysis is used very often specifically in fractures, to answer the what happened to this. And they use various visual features. Here we have an item that has 3 different fractures. In failure analysis, they can identify which fracture occurred first, and then the other fractures occurred after that. And there have also been studies that if you know the specific metallic composition of the item, and you look at the ribbing along that fracture and the spacing between that ribbing, you can predict the amount of load that was applied to that item to cause that fracture. And then we can do other things. Here we have an item that was broken, and in failure analysis, they've identified where that crack started, how that crack progressed through the item, where it ended, and how that item was moving as that crack progressed. These are very, very familiar concepts to the bloodstain pattern analyst. So what we see when looking at geology and archaeology and failure analysis is that reconstructing past events is absolutely accepted in numerous fields outside of BPA. And in every case, the reconstruction uses visual or analytical observations along with sound logic to determine the cause or the source of those observations. What is this and what caused it? But it's also essential in all conclusions, whether it's classification or a case conclusion, which are the reconstructive aspects, to be able to say, I don't know, to accept that there are some times where there's just not enough information to be certain about a conclusion. So here again is the pathway of our methodology. We've discussed this before, but now let's continue the next question. I've laid the case that this is a scientific process, pulls from other established sciences. It pulls concepts and techniques that are used in other disciplines, and it pulls those specifically for the analysis of bloodstains that are left at the scene. So it's scientific, but here's the question: is it reliable? Well, what do we mean by reliable? In a way, it kind of depends on who you ask. If you ask a statistician, an example of a statistical definition of reliability is here. It's a consistency of results as demonstrated by reproducibility or repeatability. That would be a pretty typical answer for statistical reliability. But the courts, at least in the United States, have come up with their own definition of reliability. It's similar, but it is different. Is it credible? Is the proffered evidence credible and trustworthy? So we're going to key in on two concepts: reproducibility and trustworthiness. Is your process reproducible, and can I trust that process? There's a related concept that's called validity. Reliability essentially means you get the same answer every time. But if that answer is wrong, then it's not a valid process. Validity is a process that gives you the correct or the expected outcome. So we're going to talk about reliability. In the United States, we have a Supreme Court ruling that is called the Daubert criteria for reliability. The Supreme Court stated that if scientific evidence or technical testimony is going to be provided to a jury to determine or answer a question, then the court needs to make sure that that testimony uses reliable techniques. So they established 5 different criteria. So for bloodstain pattern analysis, it's important, in fact, it's essential that I can demonstrate and articulate that BPA satisfies the Daubert criteria. So one of those, we aren't going to take these in order. First one, achieved widespread acceptance in the relevant scientific community, and BPA has satisfied that one. It was addressed in the 2009 report by the National Academy of Sciences. We had the Scientific Working Group of Bloodstain Pattern Analysis that was sponsored by the FBI. Accreditation bodies have recognized BPA as a form of testing that can be accredited on someone's scope. It has an established subcommittee on OSAC. It has an established committee on the International Association of Identification, and we have our own scientific association. So within the relevant scientific community, which is forensic science, bloodstain pattern analysis has achieved widespread acceptance. Has it been subjected to peer review? Yes, it has. There are numerous forensic journals that are peer-reviewed journals that routinely publish articles that deal directly or to some degree with bloodstain pattern analysis. In fact, as mentioned before, I'm the editor of the Journal of Bloodstain Pattern Analysis. While we don't publish a lot of articles, every article that we publish goes through a thorough blind peer review process to make sure that the work that comes out of our journal is good science. And then there are the other large and more well-known forensic science journals that all of these have published work related to BPA. And we also have multiple books as well. Regarding standards and controls, yes, they do exist. They've existed for over a decade now. In the United States, the FBI established a SWGStain, Scientific Working Group for Bloodstain Pattern Analysis, that began this process of creating guidelines and best practices and standards. That role has been replaced with the Organization of Scientific Area Committees that has a subcommittee for BPA that drafts standards and guidelines. Those standards and guidelines are published by the Academy Standards Board, the American Academy of Forensic Sciences Standards Board. So in the United States, we have standards and guidelines that are published. Outside of the United States, for those who live in the UK, the Forensic Science Regulator has published a standard on BPA. For my colleagues over in New Zealand and Australia, you have the working group on BPA that's under the National Forensic Institute. So all across the globe, we have standards that are being published to make sure that the work that's being done is good work. What about these other two concepts? What about the methodology? Has it been tested? And regarding that methodology, is there a potential to get an error rate? Well, let's see if we can answer those questions as well. And there have been multiple published studies that deal with answering that question. And we're going to go through these. A couple of them have been from the same group. But as we go into them, I have a caution. And that caution is that you don't misapply the results of these studies. That these studies provide insight into what causes errors in BPA. But please don't interpret these results as an error rate of BPA, because there are a lot of reasons why the percentage of errors may not accurately represent an error rate for the method. So there are two studies that address the role of contextual information and to determine if analysts can be negatively influenced by contextual information that is inaccurate. These two studies, one dealt with rigid, non-porous surfaces, another dealt with fabrics and clothing. And the focus of these events was very limited. Let's look at spatter-producing events and one transfer event. Both of these studies looked at well-qualified analysts, 27 to 37 analysts who were qualified and highly trained, to make sure that they had people as part of the study who knew what they were doing. But what the studies did not do was control the methodology. And they also didn't evaluate the specific methodology in causing some of the errors that were reported. So we look at the first one. This first one deals with blood stains on rigid, non-porous surfaces. 4 different spatter-producing mechanisms were studied, and across all variables, 13% of the classifications were erroneous. And what that means is that the answers that were provided by a respondent did not contain the correct answer for how the researchers knew that pattern was created. So 13% of the time, the respondents got it wrong. Well, of specific interest in this study is that sometimes those wrong answers were related to false contextual information. So this study also makes sure to be clear about not overextending the impact of this 13% erroneous rate. They say, had other more easily recognized patterns, such as drip stains, drip patterns, transferred blood, and saturation patterns, been included, the proportion of correct classifications may have been higher, which would have meant that the erroneous classifications would have been lower. And we're going to see that there's evidence that supports that. The second study that was done by this group, instead of looking at rigid, non-porous surfaces, it looked at fabric and clothing. And here, 4 spatter-producing events, mechanisms were investigated, as well as a transfer mechanism. And overall, 23% of the time, the answers were wrong. Now, this is 10% higher than what we had before. So the researchers dug into it a little bit to see why the number of erroneous conclusions was higher. And what they found is over 51% of the time that an erroneous conclusion was provided, it was all the same pattern type. So they wondered if the higher rate of false conclusions was a function of their study design. So they state that if you can argue that it's a function of the study design, then an overall rate is 14%, that may be more appropriate. So they admit that perhaps the rate of erroneous conclusions is actually 14% rather than 23%. There was also a study that was done on just general pattern recognition. This here studied the education and training differences between respondents. A single pattern was provided. That pattern was a cast-off pattern. And respondents were divided into 2 groups: the experts who had advanced training and were court qualified, and then the novices. The novices were those who had no training or only a 40-hour basic training. And what they found, we'll get to that. This is this study here. This is the one that deals with the training and experience. And what they found in that study is that for the experts, 4% of the classifications were erroneous when looking at this cast-off. And for the novices, the ones who had limited training, they got it wrong almost 50% of the time, 44%. Now, going back to this general pattern recognition, I apologize about getting the slides mixed up. Here we had a study that didn't just look at spatter-producing mechanisms. It looked at 10 different patterns that were drips and transfers and spatters. So a much larger variation of pattern types. And this looked at those who were self-proclaimed experts in BPA and those who they knew were novices. For the experts, 2% of the time, they got the wrong conclusion. But for the non-experts, the false rate was 79%. So here we have a difference on another difference on training and education, but it's looking at all different pattern types. We can see that when all different pattern types are examined, the rate of false conclusions was 2%. Another thing that we can look at is proficiency tests. There are 2 companies that provide proficiency tests for bloodstain pattern analysis. We can look at their results. They publish the results that are provided by the test takers, as well as the manufacturer statement, which is how was this pattern made. And we can evaluate those to determine how many times the test takers got the answer wrong. So for collaborative testing services, from 2005 to 2019, over 11,000 responses were submitted. And of those 11,000 responses, the correct classification was provided 94% of the time. Now, if we break that down into spatter-producing patterns and non-spatter-producing patterns, we get a little more information. For spatter-containing patterns, the correct conclusion was provided 90% of the time. If it was a non-spatter-producing pattern, that correct conclusion was provided 98% of the time. Forensic Assurance is a newer company that has been issuing a proficiency test. We have 3 years worth of data, 300 respondents, and the results are similar. The correct classification was provided 98% of the time. And the spatter and non-spatter classifications are very similar. But the challenge in using these numbers is we don't know who was taking the test. We don't know if the person was qualified. We don't even know if the person was doing bloodstain work. As long as you paid for the test, the company sent you the test. And we also don't know how many times those conclusions were subjected to peer review before they were being submitted. So there's a lot of unknowns, but this gives us a very foggy look into the number of times an incorrect conclusion is issued. So as we summarize those, what we can see for all pattern types is that in one study, it was 2% errors. For experts, for non-experts, that error percentage was 79. In proficiency tests, for both of them are either 2 to 6%. When we look at spatter-only patterns, that range is 13 to 23% in one group of studies. But when we look at a second study that splits it between experts and novices, it's 4% for the experts and 44% for the novices. So this gives us an insight in some of the things that may lead to erroneous conclusions. But what we don't know from any of these published studies is the specific method that was used by the participant. When did they use contextual information? How did they classify their patterns? Was it based on, yeah, it looks like that? Or how can I exclude other patterns and see what's left? What criteria did they use for classification? And did they submit those classifications to peer review or technical review before providing the final answer? Because understanding the method is absolutely essential in determining the source of the errors. Because when it comes to the method, and I'm going to emphasize methods over and over again, a method is no good, even it's when rooted in a broader scientific method, if it's only good if it is consistently applied the same time every time. So if you have a method and you don't really follow it, that method is just a piece of paper. It's not helping you at all. Individuals who have no established approach or who do not use the same approach are not following a reliable process. So those are attendees out there who are skeptics, or maybe even those who are attorneys, and trying to evaluate the conclusions that are offered. This is a critical question. What method did you use to come up with this conclusion? If they cannot provide an established method, or they just say, well, I just use my training and experience, well, that's not a method, and that doesn't lend to reliable results. Think about making a pie. If I want to make an apple pie, and I want to make a good apple pie every single time, what do I need to do? Well, I need to have the knowledge and skills, but I also need to follow a recipe every single time. If I had no recipe and I just did whatever came to mind as I did the work, I'm not going to have the same pie come out every time I try. That analogy applies to BPA. What method are you using, and are you consistently applying it? And that helps us when we do root cause analysis. And when we recognize that a mistake has been made, we need to take a step back and figure out why that mistake was made. And this is a process that my laboratory does. It's a root cause analysis, and look at what caused that error and what can we do to make sure that that error doesn't occur again, or minimize the potential that that error will occur again. And the first step in root cause analysis is to ask, what method were you using? Once we identify the method, then there's 4 possible answers onto what caused that error. One is that the method is reliable, but the analyst is just not qualified. This is a good recipe for an apple pie, but that person is not a cook, and that's why it messed up. Another way is that the method is reliable, the analyst is qualified, but they did not properly apply the method. They made a mistake in the application of the science. The science is valid, the method is valid, the analyst is qualified, but the analyst made a mistake. Another way is that the analyst is qualified, but the method is not reliable. Now, we need to take a little bit of caution with this, is that in bloodstain pattern analysis, we are finding that there are gaps in our knowledge, that there are things that we don't understand as much as we would like to. And even though the general approach of BPA is valid, if we find one of those knowledge gaps, we found a hole in our method. And the more research that we do, the more experience that we get in BPA, we can help fill in those holes. But it may be that that specific evidence or that specific pattern has caused issues with the method. Or it may be that everything went wrong, that the analyst isn't qualified and the method is not reliable. But what concerns me is that there is this notion to automatically jump to a specific reason why a mistake was made. And that is that the method is junk, that the whole science is bad. And often when we do that, we're really not doing a true root cause analysis. In my personal studies, as I look at the errors that have occurred in my discipline, many times it is because the analyst made a mistake. The method was good, but either the analyst misapplied the method or the individual was not properly trained and qualified. And that's why the error occurred. Errors from unqualified analysts are things that we need to address. And as we discussed previously, that there are at least 2 studies that demonstrate untrained or undertrained individuals have a higher percentage of erroneous conclusions for classifications. So I want to emphasize the second point, that if you have just attended a 40-hour introductory workshop, that does not qualify you to do bloodstain pattern analysis. And I don't care what degree you have. I don't care if you have a bachelor's or a master's or a PhD. If you have just taken one 40-hour workshop on BPA, you are not qualified to do the work. And the studies that we've gone over have demonstrated that those who just have a basic introduction to BPA are at the greatest risk of making errors in classifications and case conclusions. So how does one become competent? The American Standards Board has issued a standard or a guideline for the training of bloodstain pattern analysts. And this was started over a decade ago in swig stain and has been picked up by OSAC here in the United States, that we have consistently said 40 hours of training is not enough, that you have to have a defined program. Now we are recommending a 4-year degree, but besides that, what is your program? Does your training program have written tests? Does it have practical exercises? Does it have competency testing? And are you being guided by a qualified mentor? Now, for those who are in the legal community, this is something that we kind of need your help on, because the legal definition of competency is much, much lower than this. My understanding after talking with a judge is that according to the courts, at least in the United States, all you have to demonstrate is that a person has more knowledge and training than an average person to be determined an expert. So a 40-hour basic is something that the common public does not take, so that qualifies them with that very, very low threshold. The bloodstain community has consistently said for numerous years that is not enough. Yes, it meets that very low threshold that's established by the courts, but that threshold is opening up the potential for the court to qualify people who are at a high risk of making erroneous conclusions. And along these lines, let's be clear about something. This here is the certificate that I got when I took my 40-hour basic. For those of you who have taken an introductory course, you have probably left that course with a similar certificate. This does not certify you to do bloodstain pattern analysis. I'm going to emphasize that again. If you get a certificate of attendance, you are not a certified bloodstain pattern analyst. If you look at the reading, it simply says, Mr. Morris successfully completed a course of instruction. I was there for 5 days. I did all the work. I got it, took a test, and I'm good to go. I completed the course. It does not say I'm good to do casework. The International Association issues a certificate in bloodstain pattern analysis, and this is what that certificate issues. It looks like with this, I had to demonstrate that I had taken a basic course. I had to take hundreds of hours of additional training. I had to submit to a test that was provided, a written test, as well as a practical exam, and I had to pass that. And every 5 years, I have to recertify by demonstrating activity in the discipline of BPA, as well as demonstrating proficiency and competency by passing a practical test. That is certification. And for those of you in the legal community who are on board, there are less than 50 individuals internationally who are certified bloodstain pattern analysts through the International Association of Identification. So what about an unreliable method? What makes a method unreliable? Well, if you have inconsistent or non-existent criteria for your conclusions, That's not a reliable method. If I ask someone, how did you get to this? How did you get from the pattern to this conclusion? If they say, well, that's based on my experience in education, that's not a method. That's not a method at all. That's not rooted in a scientific process, and that does not account for uncertainty. What we found is that methods that were previously considered reliable will often need to be modified upon new information. And this is the wonders of those of you who are on the research committee. You are consistently probing new areas of bloodstain research and answering those questions that we've needed answered. You've filled in those gaps. You're studying things that we've needed to have answered for years, and I thank you for that. These are the previous known and maybe even unknown holes that have resulted in errors in the past. And for those of you who are researching BPA actively, please continue to do so. The method is absolutely critical. If you ask someone, how did you get here? And they say, well, I did bloodstain pattern analysis. That is not a method. That is a science. It's not the method. I was previously a drug chemist. And if I went to testify on my conclusions and they asked, Mr. Morris, what method did you use to come to this conclusion? If my answer is, I use drug chemistry, well, that's an unacceptable answer. Mr. Morris, what method did you use? I did color tests. I also did gas chromatography and mass spectrometry. Well, now we're getting into the method. Bloodstain pattern analysis is a science that uses methods to come to those conclusions. And determining the reliability of the conclusions means an evaluation of the methods. Here are two methods that could both fall under the larger umbrella of bloodstain pattern analysis. But as you look at these methods, if you look at the information that is used and the order of the information, they are not the same. And because they are not the same, they are not the same method. And because they are not the same method, then they may not have the same reliability. Even though both of them are used to perform bloodstain pattern analysis, they are not equivalent. And because they're not equivalent, the reliability of those two methods can be completely different. So let's answer this statement here. The uncertainties associated with bloodstain pattern analysis are enormous. There are times where I walk into a crime scene and I completely agree with this question, that I am very uncertain about what occurred here. And I am not going to give a lot of very specific conclusions and interpretations. We need to embrace uncertainty. We need to embrace the times where we simply do not know the answer. I want you to look at this pattern here. And I want you to look at the features of this pattern. And we're going to ask the question of what created that pattern. Now, as we look at this, we're going to see circular to elliptical stains. And what patterns do we know consist of circular to elliptical stains? Well, here's a variety of those. And these are various different mechanisms. So initially, all of these have to be included, but we can start cutting some of these out based on various features. But there are some of them we may not be able to cut out. And that's because there is uncertainty. So if you look at this pattern, what conclusion would you think is good for this? What would actually be an incorrect conclusion for this pattern? What is wrong? If your answer to this pattern was that it was an impact pattern, you were incorrect. This pattern is not an impact pattern. It looks like a radiating distribution of circular to elliptical stains, and we almost always jump to that conclusion, but there are features that point away from this. This is actually a cast-off pattern. I know it's a cast-off pattern because I made it. And when I made it, I thought this would be a problem if this were at a scene. For me, the correct conclusion that I'm going to report out is these are spatter stains. And that's as far as I'm going to go. Now, I might be able to exclude, you know, that this isn't a drip pattern, that this is satellite stains from a drip pattern, or if there's no gunshot injuries, it's not a forward spatter or back spatter. But in my report, I'm going to call these spatter stains because I just don't think there's sufficient evidence to be more specific than that. So what we have learned here is that we need to be asking the question, when errors occur, what method was being used? When we look at the method, then that will go a long way in determining what caused that error. So again, as we look at the Daubert criteria for reliability, I want you to take a close look at this. And the way I understand this is that analyst competency and method reliability are completely separate issues. That if we do that root cause analysis and we determine that the individual who made the conclusion was not qualified, then method reliability is off the table because the issue is with the person. They just were not qualified to do the work. So analyst competency and method reliability, or the reliability of the science, are completely separate. But this distinction is not often clear or not often made at all when people claim that bloodstain pattern analysis is junk science. They claim that a mistake was made, and it may be a horrendous mistake. And they say that mistake means the BPA is junk science. No, it doesn't. Why was that mistake made? Did the person know what they were doing? So as we move forward, what do we do now? I hope that I have demonstrated that bloodstain pattern analysis rests on a solid scientific foundation. But even though it rests on that solid scientific foundation, the perceived validity by the courts and by the public of this science is largely dependent on the people who do the work. It's dependent that the people who do the work are responsible and ethical in what they do, and that they are trained to competency. If you are not trained to competency, please do not do casework. Please find a mentor, follow an established program, and take that time to do the training. If I wanted to perform polygraphs in the United States, it would take me 18 months of training to get into polygraphs. If it takes 18 months of training with hundreds of hours of practical experience to be certified as a polygraph examiner, why do I think that I can do bloodstain work with 40 hours of training? We can't do it reliably. So the key point, some key points are bloodstain pattern analysis, it is a science. It's not an investigative tool, and it needs to be respected and honored as a science. It can provide investigative information, but it does so in the realm of science. And if you are being trained, please do not do casework until you are trained to competency, until you have been signed off by your mentor. And also, please stay current in the field of bloodstain pattern analysis. The journal that we publish with the IABPA comes out quarterly. Every issue, we include a list of recently published articles that are related to bloodstain pattern analysis. Because for me as the editor, it's important that I let the community know about all of the work that's being done. And we don't just publish research articles from other journals. We notify people of theses and research projects that have been published that may be available to answer some of these questions that we have. Be aware of published standards. If you're in the United States, be aware of what is put out by the Academy Standards Board. Those of you who are in the UK, be aware of the Forensic Science Regulator and those who are in New Zealand and Australia, make sure you are aware of what the National Forensic Institute issues. So here are some summary thoughts. I believe and I hope that I've convinced some of you, hopefully all of you, that bloodstain pattern analysis rests on a strong scientific foundation because it pulls principles from other established sciences that have direct application to understanding bloodstains and how they're created, that the feature-based approach of classification is used routinely in other sciences, that the element-based approach to reconstructing stories is used in other sciences, that bloodstain pattern analysis satisfies the Daubert criteria for reliability. But with that said, I fully admit that more research needs to be done. And I continue to encourage our researchers to press forward with that research. I encourage the bench analysts or the field analysts to do their own research, to answer their own case questions, and then to publish that research so that the broader community can benefit. And erroneous conclusions in BPA can be from a variety of different sources, from a poor method to an inappropriately applied method or an unqualified analyst, not because the science is junk. I want to thank all of you on behalf of the International Association of Bloodstain Pattern Analysts for setting aside a portion of your time at various points in your days, in your mornings, in your evenings, in the middle of your day, that you took the time to attend this webinar. We truly appreciate that. If you have questions or if you'd like some more information, my contact information, at least my contact information is there. You can email me at a couple of different addresses. You can go to the IABPA website and submit questions or comments there. Please don't leave. We've got a couple other things that we would like to do. We mentioned in the announcements that we were going to provide a certificate for attendees. So I'm going to put a poll out, and it asks two questions. Are you interested? Well, before we do that, Casper, let's if you could talk about the upcoming conference that we have next month.
Of course, Jeremy. So as at the beginning, our president said that there will be a conference, and I have a great honor to be a chairman of this conference. So it is our great pleasure and honor to invite you to this conference. It will be between 16 and 20 of November. So it's going to be on next month, and it will be totally in a virtual world. And I think it will be very interesting event. It will cost $30. So it is in reach most of us. I think that presenters so far are astonishing. The presentations will be very interesting, and it will be 5 days of full BPA work, and we really cannot wait to see you, all of you, on this conference. And as you can see, you can pretty easy register yourself by our website, iabpa.org. Every detail is over there. So you are more than welcome. Thank you.
Thank you, Casper. I appreciate that. And I kind of alluded to this earlier. We've made an offer to provide certificates for attending this webinar. I'm going to launch a poll, and it's a very quick and simple poll with 2 questions. Are you interested in attending the virtual conference that Casper discussed next month? And would you like a certificate for attending this specific webinar? And if you like a certificate, please go ahead and answer yes. And at some point in the near future, we will have that certificate sent your way. And if you would like to attend the conference or would like more information about the conference, please answer yes, and we'll get you on a mailing list. Please visit the IABPA website for more information. Registration is also live. And so I'll leave this open for a few more seconds. For those of you who may not be able to see the poll, please go ahead and email me or submit a request for certificate on the IABPA website, and we will get that taken care of. So a few more seconds, and then I'll close the poll. All right, and one last thing that we wanted to do is kind of get a sense of the people who are among us. And this here's a short poll. What's your role? You know, we kind of like to see the cross-cutting of the people who are here. We've got people who are scientists. We have people who are investigators, people who are instructors. Kind of get a sense of who is with us. So if you could provide those answers, it'd be outstanding for us. And again, we cannot thank the attendees enough for setting aside some of the time to come here. All right, I'll close it in a few seconds. And these are the results of that. So you can see that we have about half of the attendees are crime scene investigators or government employees. We also have a large number of students, and welcome to our students. And thank you for your interest in forensic science. We hope you continue your studies in that. And then for those who are instructors and private and self-employed and other, again, we thank you for being here. And I also want to issue a special thank you to the skeptics who are among us. We encourage those who are skeptical of bloodstain pattern analysis to attend. It may seem odd for me as a defender of the science to thank the skeptic for being present, but I really do. I appreciate your presence here. I appreciate some of the challenges that you've issued, because we use those challenges to make us better. And I hope that we've answered some of your questions.
If I can step in here, this is Eugene. For those of you who have any issues with the poll, just go ahead to the IABPA.org website, and on the contact form, send us an email, let us know that you were here, and we'll make sure we can check that or cross-check that and then get you the certificate. Thank you.
Thank you, Eugene. This concludes our webinar for today. Thank you so much. Chelly unfortunately said that she was not able to be here for the end of the presentation, but she, as the president, wanted to thank all of you for attending, and we look forward to seeing many of you at the upcoming virtual conference in November. Thank you again, and whatever your day does, your evening does, I pray that you are safe and healthy during these times. Thank you again. Goodbye.